This week we're going to be finishing up the tissue chapter, the nervous tissue chapter by concentrating on what I'm calling excitable membranes. So first I want to just point out to you the folder for this week. When you open it up, this is what it'll look like until you finish the nervous tissue chapter assignments. It here tells you that there is an overview assignment on excitable membranes, but you won't see that the link to that assignment until you've finished part 4 of the nervous tissue chapter assignments. And so when you do see that link here, you can go in to do that. It's an overview assignment, kind of like I had that overview assignment earlier in the semester that was about the levels of organization, the first 4 chapters in the book. This one's an overview about electrical activity in the tissues that we've been studying recently. So it talks about excitation in controlling skeletal muscles and the stuff that we're going to be addressing tonight. Also in this chapter, in this folder, is the link to the nervous tissue micrographs that I was showing you last week, and the lab quiz on neuroanatomy. It kind of depends on exactly where we get to tonight, which I have a feeling I know what's going to happen, but that lab quiz is only partly over the tissue stuff that we talked about last time. It's also about the broader neuroanatomy of the nervous system. We're going to look at models of the brain, get to know how the nervous system is arranged. And I really don't think we're going to get to looking at that tonight. To talk about the excitable membrane stuff, the electrical activity. I'm pretty sure that's probably going to take most of tonight. I'm not positive about that, but I'm pretty sure it will. Or it'll take us in the second half, and I might get started talking about the rest of the neuroanatomy, but I doubt I'm going to finish it tonight. If that is the case, the due date for the neuroanatomy lab quiz right now is before next week's class. If I don't finish that material tonight, I'll change that due date to be a week later, because we'll finish the material next week, and we'll go on from there. So, but there are not any more part 1, 2, 3, and 4 assignments for this week. The assignments in last week's folder are over the chapter, which we're still continuing on. So. And those, while they're in last week's folder, they're due before next week. They've always had that due date in them. And the due date for the overview assignment we see here is also before next week. So what we talked about last week was nervous tissue. We looked at the micrographs of the nervous tissue before. Looked at the micrographs of the nervous tissue in that link I just pointed to that's in this week's folder. Now we want to get to talking about the electrical function of the system. Now, like I said before, yes.
I wanted to tell you something about that kid that I was telling you about that had the spinal injury last time.
Okay.
He typed on a computer with his right hand.
All right. Good to hear.
So because he's had movement and it says it happened, can you be paralyzed from the chest down but be able to have access to using his hand? Well,
the nerves that control the hand are actually coming out of the base of the neck. So they're coming out right here. And so these big bundles are the nerves going to the arm. So he's controlling his hands. The neurons controlling that are coming from this level right here. So if he had damage somewhere in his chest,
she said that he broke his neck. So say like it was only maybe one side that was broken.
Well,
look at that.
When you say breaking the neck, you're really talking about the bone structure there.
Yeah.
It doesn't necessarily imply that you damaged the spinal cord, and it is possible that part of it's damaged and part of it's not. And actually, I can't remember if it's next week or the week after, I'm actually going to talk about what are called hemisections, which are where half of the spinal cord is damaged.
So that could be why he has his movement to his right but not left, because it could be a half
Right.
of
We usually think of damage as being, sort of straight across, but if the trauma is coming from one side, it might damage one side more than the other.
being that he got hit by a car, I mean, it could have been just an impact from one side.
Right. So keep that in mind, and we'll be looking at the connections and pathways that control movements and how they're affected a little bit in those kinds of injuries. Okay, so the next few sections here talking about how membrane potentials work. I want to kind of preface this with a little bit of, my own experiences and learning about this, and then what I did for putting this together. When I was in graduate school, or actually I guess it was, I think I started learning about it in college, but when I really started to learn this stuff in depth, it was in graduate school. And there's really two sort of approaches to learning this material, learning the electrical activity in physiological systems. One is based on understanding electrical systems, resistance, capacitors, batteries, currents, those sorts of things. And the other is to talk about it more in a particulate manner, talking about ions and channels and extracellular and intracellular fluids. At the doctorate level, the first approach is the one that's usually used. Somebody who's gotten to the level of graduate work in neuroscience or in biology of some sort has probably studied university level physics, which gets into how electrical circuits work and capacitance and resistors and all that sort of thing, as well as higher level mathematics, calculus and beyond. I actually never went that far in physics and math. My college education was a little bit, shall we say, meandering. When I started college, I wanted to go into chemical engineering because I was working in photography and I wanted to study chemicals. Glad I didn't do that because, within a couple of decades, photography in that sense is completely gone. But then I wanted to go into American studies, sort of a specialization within history, because the college I went to was very strong in that, and I was taking a class and I really rather enjoyed it. And then I got kind of tired of that, and so I decided I wanted to go into computer science. And one of the things you have to take for computer science is higher level math, and I did really poorly in calculus. I would have failed calculus, except that the professor actually rounded everybody's grade up by one letter, so I went from an F to D, which I really didn't deserve. I completely failed that class, but he had his reasons for doing that. So I was able to squeak by with some credit for having completed calculus, but I didn't really know calculus. And so I was like, well, I'm not going to go into computer science. I don't have the math to do that. So it got to the point where I had to declare a major. And my dad's a psychiatrist. And at the time, my parents had divorced, I guess when I started high school. But at the time, my dad had been living with his girlfriend, which just really sounds strange for a man of his age. But the woman he was with after he divorced my mother, and she was a psychologist. So when I lived with them, it was sort of like this, salon where we were talking about the brain and psychology and that sort of thing. So I'm like, well, I got to pick a major somewhere. Why don't I go ahead and go with psychology? I know that stuff pretty well already, so I went into it. And in doing that, I decided that I liked best the biological side of things, how the brain works. So I went into experimental psychology. And I designed my major actually by including a minor in biology to essentially be a neuroscience major. Although back then in college, they didn't have majors that were that particular. It was, I was a psychology major with a bio minor. But I took classes sort of to get at understanding the brain and communication a little bit better. And in doing that, I didn't have to take more than a semester of physics. So I took the physics where you learn about, Newton's laws of motion and that kind of thing. But I never got around to taking the electrical stuff. So I get to graduate school, and I know the basics of how this stuff works from my biopsychology classes in college, but it really took it to a whole new level. And I struggled with it the whole time, and I hated it because it didn't make sense to me. And since then, since I was in graduate school, I've been teaching in a lot of different situations, teaching to a lot of different levels of students. And I find that coming from a biology perspective, understanding how this works from a particle point of view, in that ions are particles that are moving in and out of cells, makes a lot more sense than talking about it in terms of batteries and resistance and capacitance and all that sort of thing. I'll mention a few of those words just so that you understand why that's part of it, but I want to get at more with the particle side of things. And so as I was writing this stuff, I wanted to concentrate on that approach to things. And I also wanted to try to, get at it in a sort of more straightforward or quicker way, get right to the heart of what's going on here. I don't really feel I did that great of a job in writing the book here, and I really want to revise this part of the chapter, which doesn't help you a whole lot. But in presenting this tonight, I kind of want to try to make up for some of the shortcomings here. So as I'm presenting this stuff, I will be working out of the material in the book, but I'm going to jump around a little bit because I think I needed to rearrange stuff on the chapter a little bit. And there are a couple of things that are missing. I don't have good pictures for certain things. So I'll try to represent that for you. I might have to pull up pictures off the internet somewhere else to demonstrate things. Now, part of what sort of, I don't know if inspired is the right word, but guided me into how I wanted to present this stuff came from a textbook I used to use teaching that had this sort of transitional section between the anatomy of nervous tissue and the physiology of nervous tissue, setting up a picture kind of like this, looking at all of the players in a rather complex circuit. I really struggled with the, I really struggled with the art editor to get this picture the way I wanted it to. And now that I'm using it, I'm actually a little upset. It didn't quite do everything I wanted to. And that's just a matter of how it had to be centered on the screen. What we're looking at here is the brain and a cross-section of the spinal cord, and then obviously the hand out in the periphery. Really, the brain and the spinal cord should be lined up together, and because they're not, there's actually something in this that isn't as obvious as it should be. So let me just point out to you, this is the midline of the brain, and this is the midline of the spinal cord. If they were in fact lined up together, these two lines, the red and the blue line here, that kind of go over to the side, really should be going straight up. And the angle that we see here to make them go from this part of the picture to this part of the picture is very misleading, because in fact, these two lines would really be coming from the other side of the brain. And if you really look closely, the connections are correct how they're laid out here, but this little funny jog right here kind of obscures that fact, okay? So I would kind of like to fix the picture in that sense. The other thing that I never even noticed while I was working with the art editors, that's the wrong hand, okay? Which isn't terribly important, except that things are labeled as right and left. Really what we should be looking at is the brain, like we're looking at my head right here, and the spinal cord would be oriented kind of like my spinal cord is here, and so it should be this hand, okay? But in fact, this is the mirror image of what I'm holding out. So this is this hand here, okay? But really the idea is that it's the hand on this side of the body connected to this side of the spinal cord, connected to this side of the brain, okay? So just ignore the fact that this is actually the wrong hand in the picture, but it's in the right part of the picture to represent that side of the body. So we'll just pretend that this is the right hand and not the left hand, and we'll be happy. Now the point of this picture is to show how all of these cells in the tissue, the neurons in the tissue, are integrating together to provide the full physiological relationship within a complex circuit. So we have sensory endings that are receiving a stimulus. So that's the sensory function that I mentioned last time. And that's being carried into the central nervous system, where other cells start to pick up that information and send it around to different places, which is the integration aspect of what's going on. And then there's an output that controls the movement of muscles. Now, I can't remember offhand exactly what my original intention for this picture was supposed to be. I think I might have been working with touching a hot stove or something like that, but that was a little bit too reflexive for what I wanted to do. And so the art editor suggested this sort of thing, sensing the temperature of water in a shower. And so the response, the movement that has to be coming from this isn't really obvious from this picture, but let's say you've put your hand in here, and based on whatever temperature the water is, you pull it back and you adjust the temperature of the knobs. Not really depicted that way, but that's kind of what I was going for here. Now, in this picture, the sensory endings here could be anywhere in the skin that's being hit by the water, but we just put it in the fingertip. Those are the dendrites of a particular neuron. And that neuron, from that dendrite, those dendrites, an axon extends through the hand and the arm and eventually comes to the spinal column. And right before the spinal cord, there's a ganglion, the dorsal root ganglion, which is what I was showing you last time with the unipolar cell, where the cell body is located. And that cell body is what this axon is attached to. The axon continues on through the ganglion and then enters into the posterior or dorsal side of the spinal cord. It enters into white matter in the spinal cord, and then what this little bend here is supposed to signify is that it's turning to move to go up the spinal cord to the brain. Okay. Now, like I said, the way that this got set up with the brain off center from the spinal cord, the blue line goes off to the side here, when really what's happening is it's turning and crossing midline to go up the other side of the brain. So if this, the midline of the brain here correspond to the midline of the spinal cord. From here to there, it would be going from the right side of the spinal cord eventually to the left side of the brain. That's really what's happening there. Okay. Now, the axon that we've been following all along from the fingertip here finally synapses on the next neuron in the chain. Okay, and this neuron's in the brainstem. And really, this neuron should be on the other side of the midline, but it's just sort of floating around the white space the way this is drawn. Okay, and from here across midline, this is where the information from the right side of the body ends up going to the left side of the brain. Okay, and its axon then synapses here, and then this third neuron sends its axon up to the cortex of the brain. Now, there's no target neuron represented here because from this point on for a little while, it gets way too complicated to represent in a picture here. So the idea is the sensory information is coming in along all of these blue colored neurons. And there are 3 in the circuit going from the periphery where the sensation is originally encountered to the surface of the brain where that sensation becomes a conscious perception. And then from there on, there are lots of neurons that are involved in the integration of putting all that information together and deciding what to do with it is more complex than we want to represent here. But what ends up happening is the information gets to this part of the brain where it initiates the signal that's going to cause some sort of motor response, some sort of movement. And so we have a neuron whose cell body's up here in the brain. Its axon in red now goes down. And again, because of these 2 being off center, it looks like it's turning this way, but really it's going from the left side of the brain to the right side of the spinal cord. It finally synapses on a neuron in the gray matter of the spinal cord, which its axon then goes out through the nerve to make connections to muscles by neuromuscular junctions in the hand, which might bend the fingers or pull the hand back or whatever from the water that's being tested here. This neuron here in this gray matter of the spinal cord, that's the neuron that we were looking at last time. So when I first started showing you cells in the micrographs, that first type of cell that we were looking at was this type of neuron right here. It's responsible for controlling muscle cells. Okay. It's the neuron that is in charge of a motor unit. Okay. So they're not depicting it as synapsing on specific muscle fibers here. It's just going out into the hand where it controls muscles. But that's what it's responsible for. It's controlling muscle contraction. So it's the motor unit neuron that we met in the chapter on muscle tissue. So that's the players, the cells that are involved in the process of receiving information, sending it into the central nervous system in the brain, where we process that information, then responding with a motor output kind of response. Okay. The point here is to kind of set up the idea of the electrical signaling. And so these arrows that we see represent the communication that's taking place. Some of these are electrical signals, some of these are chemical signals. But starting here, information flows along this axon to its synapse here, and then it starts a change in this neuron, which travels along its axon to this point, which then causes a change in this neuron, and it sends a signal along there. And then the same sort of thing happens coming out from the brain to to the skeletal muscle there. So what I want to talk about are the signals that we're seeing here. And to describe that, I need to introduce some terms to you. First is membrane potential. The membrane potential is the distribution of charge across the membrane. Now, the word potential here is used kind of in the sense of potential energy. This is by distributing charge across the membrane, we're setting up a well, there's really no way to say this clearly without going to the more electrically based aspect of this. So by distributing charge across the membrane, we're setting up a battery that we can use energy from. Okay. So I have here a battery, copper top battery, which is commonly found in electronics, TV remotes, whatever it might be, a AA battery. It has a positive end and a negative end. Okay, so there's charge distributed across this battery. If we look at the membrane of a cell, there's charge distributed across that. It has a positive side and a negative side. And just like we can complete an electrical circuit by putting this battery into a electronic device, we can allow the charge that's distributed across the membrane to flow through a circuit, through a pathway, and we can get some energy or a useful electrical signal out of that. Okay. Yes?
With the membrane potential, are you talking about the actual membrane, the lipid bilayer? Or are
Yes.
you talking about the inside or the outside of the cell?
We're talking about distribution of charge across the membrane from the outside to the inside. Okay. And so, let's see. This picture depicts it. Okay. So here we have our phospholipid bilayer membrane. And charge is distributed so that one side is negative compared to the other side, or one side is positive compared to the other side. There's a positive charge on one side and there's a negative charge on the other. Now, part of what this picture is depicting is how we measure that charge. So this little needle here that's piercing through the membrane contains a recording electrode, microelectrode. Now, the way this is drawn is a little bit oversimplified. It's not going to go from exactly right outside the membrane to just inside. It'll go just inside, but the other end of this electrode is actually going to be physically far away from the cell membrane. When you're measuring this, you're measuring it usually in a Petri dish where you have the cell isolated and it's suspended in some sort of nutrient bath. The electrode pierces into the cell, the needle point goes into the cell, but it's sticking out of the Petri dish. You can see the other end. Can't even see the cell, it's microscopic. But the microelectrode that we're talking about here is huge. So this end of it is actually far outside of the solution that we're talking about. It's not just floating in the extracellular fluid. And the microelectrode, the wire that's inside this needle, is attached to a voltmeter, represented here. And the voltmeter has also attached to it another electrode, not in the sense of a needle microelectrode, but just the end of a wire that's in the extracellular fluid. And so by comparing the charge that we record with the reference electrode to the charge that we record at the tip of needle microelectrode, we can see what that distribution of charge is across the membrane. Okay. Now, the convention is to compare the inside to the outside. So outside would be like thinking of that's the zero charge. And the inside is negative 70 millivolts less than the outside. Okay. We could just as easily say that the inside is zero and the outside is positive 70 millivolts in comparison. Okay. There's a difference of 70 millivolts across the two, inside negative relative to outside. So when we say that we're measuring negative 70 millivolts, that really means that what we're measuring inside is 70 millivolts more negative than what we measure at the reference electrode. That could be 100 millivolts versus 30 millivolts, or it could be 250 millivolts versus 180 millivolts, or it could be 20 millivolts versus negative 50 millivolts. We're not talking about the actual charge at that electrode, but just the difference between them. That's what distribution is talking about. How much charge is spread across that membrane. Okay. So let me get back to this battery for a second. This is measured in millivolts. Okay. There's a 70 millivolt difference that we see across the membrane there. How many volts do we see distributed across a battery like this AA battery that I have here? Yeah. Okay. This isn't a lesson for all of us in how you're not very observant. Back when I was talking about microscopes, I also asked everybody if they knew what year this building was built in. You remember that? Okay. Nobody pays attention to this. There's a marker on the outside of this building that says 1987, which is when this building was built. Nobody notices that. Okay. I'm sure everybody has used a AA battery in their life, but you've never looked at the battery to see how much charge it actually has. Okay. And right here, it says AA, and then it says MN 1500, which must be Duracell's particular name for that. It also says LR6, which must be another name for this type of battery. And then it says 1.5 volts. Okay. Nobody ever pays attention to that, but these are 1.5 volt batteries. Okay. Now, if I had instead started with this battery, you would have been able to answer that. How many volts is in this battery right here? 9. It's a 9-volt battery. Okay. But a millivolt is 1,000th of a volt. Okay, so 1.5 volts is 1,500 millivolts. 9 volts is 9,000 millivolts. Okay. The distribution of charge here is very, very small, 70 millivolts. Negative inside compared to outside. We're not powering something like we're powering an electronic device with one of those batteries, but it's we can use the metaphor of the battery, so to speak, to describe what's happening here, because it is that sort of thing. It's distribution of charge from a positive to a negative side. Okay. Now, I went down a little bit to show you this picture. What I skipped over was a little bit more detail about the membrane. Okay, so here's a picture of the membrane, and membranes actually have various proteins associated with them. Some of them cross over the entire membrane. Some of them are only found on one side. And some of those that cross through the entire membrane have an opening through them called a pore, and we call that a channel protein. And it allows things to cross from one side of the membrane to the other. Now, what we're interested in seeing cross the membrane are ions. And the ions that we're most concerned with are sodium, potassium, calcium, chloride, those kinds of things. They're fairly small ions, but the next few pictures here, which in retrospect I might have presented a little bit differently, show different types of channels. So let me talk about these a little bit. So the first type, which we've actually met before, is called a ligand-gated channel. Let me go back and point that out to you. Pretty cool. Ligand-gated channel. The word ligand, means something that binds to the receptor. Okay? Now, we're talking about neurons here. So the ligand is a neurotransmitter. Okay? But we want to use a more general term like ligand because in other situations, there are other things that bind to receptors. Okay? So ligand is just a thing that binds to a receptor. And in this picture, we have represented by a red triangle, the acetylcholine molecule. When two acetylcholine molecules bind to this receptor, there are two spots there, the pore opens up, okay? And in this picture, we see sodium outside the cell, calcium outside the cell, and potassium inside the cell. When acetylcholine binds to its receptor, which is a ligand-gated channel, sodium and calcium will move into the cell down their concentration gradient, and potassium will move out of the cell down its concentration gradient. You're always going to see movement of ions across or through channels based on their concentration gradient. Wherever there's more ions, the ions will move to where there's less ions, just a difference in concentration. There's more sodium and calcium outside of a cell, more potassium inside. So they're going to move in that direction. Okay. Now, we've met this before. This was the acetylcholine receptor that was at the neuromuscular junction that we talked about a couple of weeks ago now, I guess. And all we really cared about there was the sodium that was moving in. Okay. But in fact, it's a sort of broadly permeable channel, and any positively charged ion can cross over. Okay? But that ion's going to move down its concentration gradient, depending on where it is. Okay. Another type of channel is called the mechanically gated channel. And here we see sort of a schematic representation of one. We can see the pore, which looks like it's wide open, but instead there's sort of a lid on top, kind of like a garbage can. Mechanically gated means that a physical force will pop the lid open on this. Okay, we see mechanically gated channels in places like in the skin where we're sensing touch, pressure, vibration, all those sorts of things. The physical stimulus of what's actually interacting with our skin can make this kind of channel open up. Okay, and again, this channel shows multiple things going through it. I can't remember now. It's been a while since I worked on all this stuff to make the book. I don't remember why calcium's concentration gradient's shown going out. It might be because this example is from a special kind of cell where that's true, but we're not really going to deal with calcium a whole lot in terms of this. It's a player in some circumstances, but sodium is usually the more important one that we're talking about here. And so some physical change to the cell, like pressure that physically deforms the shape of the cell, will cause this to pop open and the ions can move across. Okay. The next type is called a voltage-gated channel. And when it's closed, the gate is this little ball and chain looking thing here that's plugging the inside of the channel. The pore, the rest of the way, looks fairly open. It's just that it's blocked on this side. The way this is depicted, there's two little pieces of the protein sticking out a little bit on either side. that we can also see in here, they're holding the ball part of this ball and chain in place. Okay. Now this is kind of schematic. This isn't necessarily what these things actually look like, but it's sort of depicting the process here. So the ball part is held in place because these other two things are in there holding on to it. Voltage gated means when the distribution of charge across the membrane changes, the shape of the protein will change also. And what we see is the two things on either side, which are holding the ball in place, they move and they release the ball so it opens up. And now the ions can move across. Now in this example, the ions that we see on the inside are not labeled, but it's the same color that's used for the potassium in an earlier picture. Potassium is inside the cell. And so when the ball and chain part opens up, the ion will leave the cell down its concentration gradient like we see here. Down at the bottom, it says, in this picture, the membrane is at -70 millivolts, which was kind of like the picture I just showed you. If it changes, in this case, to -50 millivolts, so the inside becomes a little less negative than it was before, that will cause the protein to change shape and release the gate, and the ions can then leave. Okay. This is a voltage-gated channel. It's dependent on changes in membrane voltage. And we're actually going to be looking at the voltage-gated channels mostly today, because they're the big players in what's going on here. Because the membrane potential I've been talking about here, as the electrical signal occurs, it's going to be about the change of that distribution of charge or that voltage. And as it changes, voltage-sensitive channels will open up and close and that sort of thing. So those are the voltage-gated ones. There's one other one that I want to mention, and this picture is a little weird. I know where it came from, but it's just still kind of weird. It looks like here's the pore open at the top, and the wall of the channel's closed up. Kind of a weird drawing. And then what they show here is that wall kind of opens up so we can see the inside of the channel. It's a weird depiction of the opening and closing of this pore, but I actually know sort of where the artist got the concept from, and it's not perfect, but it makes the point, I think. These are what are called leakage channels, which is that they're not gated. They let things leak out of the cell. They're not always open like we see here. Sometimes they're closed, but they just randomly open and close. And how fast they open and close really determines how much of the ion will leak across the membrane. Okay. So here again, we have, in this case, potassium ions inside the cell, and when it's open, they'll leak out of the cell, again, down their concentration gradient. Okay. Now, the leakage channels are kind of weird because we don't have any control over them, but they are important to understanding something about what's happening here. Those types of channels, and back up here, are what we have going across the membrane to allow for changes in the membrane potential. Okay. So back to membrane potential. Its definition is just the distribution of charge that we can measure across there, like with this voltmeter. Okay. I thought there was a picture here, but I guess it's just explaining the text. The membrane potential, when the cell is at rest, is called the resting membrane potential. Okay, this is the distribution of charge, which is voltage, across the membrane at rest, which really isn't anything different from the membrane potential here, except that the point is that the membrane potential can change. So the starting point is when the cell's at rest. Okay, and at rest, the voltage is negative 70 millivolts. Okay, again, that's negative inside compared to outside. And so this is the starting point that we're going to look at. The membrane potential will change, generating the electrical signal that we're going to work with. Okay. Now, before I get into that, actually, I think there's a video I want to show you. Huh. I thought there was another video by the same group that did the nice ones of the muscle. I don't think this is it. Well, that's obviously not that. That's an ad. Okay, well, let's just see where this goes. Excuse the 4 seconds of the ad that we have to go through here. All right. That was weird. So I might stop playing this if I don't want to continue on, but let's see what happens.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membrane. This potential, called resting potential, is always negative inside the cell and ranges from negative 40 to negative 90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold. And an action. Okay,
this is really getting away from what I wanted to talk about.
Proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium
okay.
ions.
Let me back up there for a second.
Cytoplasm with various ions dissolved.
Okay, so I skipped over a little bit because it was kind of getting ahead of itself. But here's where it actually gets to what's going on with the resting membrane potential. So what it's saying here, and it's going to kind of start up in the middle of the statement. I'm not going to try to find the exact starting of it. But what they're showing here is inside the cell, these little dots and then these larger things are ions and proteins. And I think what it's about to say is at least the proteins have a very negative charge to them.
Hence, the neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of potassium ions initially inside a neuron must leave to create a resting potential of negative 60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
Okay, so part of what I skipped over there was where I started talking about threshold is also going to start to define the action potential, which we'll get to a little later. But where I went forward there, where it started talking about it, that was a good explanation of what was going on. They oversimplified it just a little bit, but it was pretty good. I don't have this picture here. Okay, that's really, okay. One of the shortcomings of what I've done here is I don't have a good picture depicting the ions that are inside and outside the cell. So outside the cell, we have a high concentration of sodium ions. It's about 10 times more sodium ions outside the cell than inside the cell. And then inside the cell, we have potassium ions. I think it's about 5 or 6 times more concentrated. I don't remember. It's not as concentrated as sodium, but it is there. Inside the cell, we also have these negatively charged proteins, which the video just showed you. There are potassium leak channels, and so potassium will leak out of the cell, taking its positive charge with it, making the inside more negative, largely because of those negative proteins in there. What they were saying about the electrical force, that there's a balance with attraction pulling the positive potassium back in and it's leaking out also, that's the oversimplification. It's not exactly how it works, but what that's getting at is what's called the equilibrium potential. Which is the distribution, or I should, up here when I was defining the membrane, resting membrane potential, distribution of charge, I put in parentheses voltage. And so just so I don't have to keep typing distribution of charge, I'll say voltage, okay? So the equilibrium potential is the voltage, distribution of charge, across the membrane when one ion diffuses to equilibrium. The equilibrium potential of, which we can call EP, just abbreviating equilibrium potential of potassium, is negative 90 millivolts. And then the equilibrium potential of sodium is, I think, if I remember correctly, and the exact number is not that important, but I think it's positive 56 millivolts or something like that. It's very positive. Now, what this means is we have sodium stuck outside the cell, we have potassium inside the cell. If we allow the potassium to leave the cell until it's at equilibrium, which is what that video is kind of talking about, that balance of attraction and that sort of thing. If we allow potassium to actually leave the cell freely as much as it wants, so that the concentration gradient is exactly offset by that opposite attracting kind of thing, what's called the electrochemical gradient, then the membrane potential will actually go down to negative 90. If we allowed sodium to do the same thing, because sodium is more concentrated outside than inside, sodium will move into the cell and bring a positive charge inside, making the inside of the cell more and more negative. Because of the really, really strong concentration gradient, the 10-fold concentration gradient across the membrane for sodium, sodium will diffuse through to equilibrium, causing the membrane to reach 56 millivolts positive. Okay. Now, the resting membrane potential is closer to the equilibrium potential of potassium. And the reason for that is kind of what was said in the video there, that the leak channel I showed you before is really only applies to potassium. There's not really a leak channel for sodium. There is, however, a little bit of sodium that does move into the cell. Sodium is used in a lot of different ways in cells. And one way that it's used is it helps to transport glucose into a cell. So as the neuron is getting glucose for it to make ATP, it's going to be taking up a little bit of sodium. So we could call that a sodium leak, although it's not really a leak. It's just sodium being used in a different way. So as the sodium comes in with glucose and those kinds of things, it's going to want to make the membrane potential more positive, the inside of the cell more positive. It's not going to do it to equilibrium. It's never going to do it that much with the sodium moving through the little bit it does to help transport things like glucose. But it does do it enough so that the membrane potential sits at -70 and isn't going to reach equilibrium for potassium. Okay. So there is a little bit of a contribution from sodium, keeping the membrane potential a little bit higher than potassium's equilibrium potential. Okay. That might sound a little confusing at this point, but I'm trying to talk about the things that make it that contribute to the electrical signal we're going to look at. And the equilibrium potentials of potassium and sodium both play a role here. And for the resting potential, it's closer to the potassium equilibrium potential, which is very negative, even though potassium is a positive ion, because of the negative proteins that are in the cell. And then but it's not -90 because a little bit of sodium getting into the cell keeps it a little bit less negative. Okay. The real contribution of the sodium equilibrium potential will be during the action potential. That's where it really makes a difference, but it does play a role here. Now, let's see. The other type of potential I want to describe here is the action potential. And this is the, how do I like to say this? Characteristic change in voltage resulting in the nerve impulse or an electrical signal. Now, what I'm saying here, characteristic change in voltage, there's a series of events that transpire that results in the action potential. And how it works is pretty consistent. It always happens the same way. And it ends up causing a significant change in the voltage across the membrane, which will get back to its normal resting state. So it's kind of like a little switch that's flipped for a little bit and then turned off. Okay, so if I could reach the light switch easily from where I am right now, the action potential would be sort of like me just flipping the switch on for a second and flipping it off. Just a brief change, just like the voltage in the circuit going to the lights would be on for a little bit and then off for a little bit. here we're talking about the voltage across the membrane switches from negative to positive and then back to negative. Okay. So the next picture in the book is showing a graphical representation of that change in voltage, which is not that picture. Where'd the picture go? There we go. So we have the line starting off at -70. Okay, that's resting membrane potential. Something happens, and we'll get to what that something is later on, which starts to cause the membrane potential to become more positive or less negative. Okay, that's usually because sodium is entering into the cell. Okay, when it reaches a certain level, which is generally accepted to be -55 millivolts, we hit what's called threshold. And so that video I was showing you a second ago, it started to talk about threshold, which is the video I meant to show it to you to talk about resting potential, so I skipped over that. But when the cell gets away from resting potential, if it becomes more positive, it'll hit a point at -55 where suddenly the action potential has to run its course. Okay. If we change the membrane potential a little bit, but it doesn't get all the way to -55, then it'll just go back to -70. Nothing will happen. But once it hits -55, what happens now is a very rapid change in the membrane potential. When the membrane potential goes from a negative to towards 0 and even past that into the positive values, we call that depolarization. Okay, so at rest, the membrane is polarized. It's in a very negative voltage. And so if we make that move more towards 0, we're depolarizing it. Okay. Now, depolarization actually goes past 0 into a positive range, but it doesn't get as positive as it was negative before. So it becomes less negative overall. What's causing this, what's happening at -55 millivolts, is that a special kind of voltage gated channel opens up, which is sensitive to sodium, or not sensitive to sodium, but it's sensitive to voltage. It's a channel for sodium, and only sodium can cross over. It actually has 2 gates, which are under different voltage controls. At -55, what happens is one gate opens up, which we call the activation gate. So we activate the channel, and sodium can move into the cell. Now, sodium is very concentrated outside compared to inside. So it moves in very rapidly, and it gets to be a very positive charge in there. This is where that sodium equilibrium potential is actually important. Okay. When these voltage-gated sodium channels become activated, that lets sodium rush into the cell. And it's going to rush into the cell until it reaches equilibrium. Okay. It wants to rush in the cell until it reaches equilibrium. So it wants to actually change the membrane potential from -55 all the way up to its equilibrium potential, which I think is +56. The thing is that it doesn't get past +30 because that voltage-gated channel has 2 gates. There's the activation gate that opens at -55 and lets sodium come into the cell. And then there's also an inactivation gate that closes when it reaches +30. Okay. Now, having 2 gates is like having 2 doors to get in and out of your house. Okay. If you have to get in or out of your house, both of the doors have to be open. If you close one of the doors, you can't get in and out of the house. Close both the doors, you can't get in and out of the house either. But only one of them has to be closed to keep things from opening. Okay. So the example I like to use, which is kind of silly, but everybody loves cats thanks to the internet nowadays. Let's imagine you have a cat and it's in your living room, and your front door opens from your living room to the outside. but you have the actual front door, the big wooden thing, and you have the storm door, screen door, whatever you happen to have, depending on the time of year. They can both be closed, okay? But if one is open and the other's closed, it's still not going to let the cat out of the house, okay? Both of those have to be open for the cat to get out of the house. So the voltage-gated sodium channel is like those two doors. It sits there with one door open. The inactivation gate is open at rest, and the activation gate is closed. At negative 55 millivolts, the activation gate opens, and so you have both doors open, and the cat can get out of the house. The sodium can get into the cell. At positive 30, the inactivation gate closes, so the storm door closes. Now, the cat's already out of the house, so it doesn't really help that much, but the 30 other cats that you have in the house can't get out now, okay? So we'll go for the crazy cat owner person there. So they can't get out because it's inactivated. The gates open and close at different times, okay? The inactivation gate stays closed until we get back down to rest, okay? Actually, towards rest. The activation gate closes again when we hit negative 55. So eventually the activation gate will close, so both will be closed, and then the inactivation gate will open, okay? So at threshold, both are open, and then the inactivation gate closes, and then the activation gate closes, and then the inactivation gate opens again. The activation gate stays closed until the next time it hits threshold. And then when it hits threshold, it opens and the cats can get out of the house again. Okay. So at threshold, the activation gate opens and sodium can rush into the cell. That'll happen until it hits positive 30 and the inactivation gate closes. Sodium stops being able to get into the cell. At that same time, voltage-gated potassium channels will open. Now, voltage-gated potassium channels are gated to negative 50 millivolts. Okay, and actually, whoops, that's not it. This voltage-gated channel I'm showing you here, this is the voltage-gated potassium channel. It's closed at negative 70. It's opened at negative 50. Okay. The thing is, what's not obvious in this picture is that it's a little bit slow to open. Okay? It takes about a half a millisecond for the voltage-gated potassium channel to open. Okay, so back here. We hit threshold at negative 55. The activation gate of the voltage-gated sodium channel opens. Very quickly, we get to negative 50. That's the voltage that tells the potassium channels to open. But they don't actually open for about a half a millisecond, which is as long as depolarization takes. So they are told to open back here, but they're slow to open, so they're not actually open until this half millisecond later, which is when depolarization is ended. So when depolarization ends, potassium channels open finally and let potassium out of the cell. Potassium has a positive charge, and so as it leaves the cell, it's going to take the part of positive charge out of the cell and repolarize, get it back down to a negative charge. Now, these channels are open, letting lots of calcium through, lots of potassium through, more than the leak channels will allow. Okay, so now we get repolarization, which is really potassium moving to equilibrium, and it actually moves closer to its equilibrium potential than we would normally expect. Okay, as it goes past negative 50 again, that's a signal for the gates to close. It's just it takes them about a half a millisecond to close, and they're not going to finally close until actually the membrane has reached this very negative value, which isn't even marked on the graph here because it doesn't really matter how negative this is. It's more negative than resting potential. It might be negative 80, it might be negative 90, whatever it is, it's more negative. That's what we call hyperpolarization. The membrane is more negative than it's supposed to be. And after the potassium channels close, the membrane gets back to its resting potential change there. Resting potential charge there. Okay. So we get back here, and then we can see another action potential take place. Okay. So what's driving all of this is voltage changes that open and close voltage gated channels. Okay. The sodium channel has two gates, one that opens at negative 55 and another one that closes at positive 30. The potassium voltage gated channel only opens and closes at negative 50, but it takes a half a millisecond for it to actually open. So it's delayed a little bit, and it finally is open once we've hit the top of depolarization, and it finally closes when we hit the bottom of hyperpolarization. Okay. But that's the potassium causing repolarization. So what I just described using this picture is sort of laid out in the text here. I have a new version of that model, of that figure that I was just showing you there. This one's labeled, and in the figure caption, it says what these different labels are getting at. But so here's the one we're just looking at, and the new one just has some labels attached to it. So in the figure caption, it just says, number one, it's at rest. Number two, a stimulus is applied, which gets it to threshold. Number three is when the voltage rises, the depolarization. And number four is when the voltage falls, or repolarization. Number five is when it's at the end of the action potential, when it's hyperpolarized, and then it returns back to rest. So the one thing that's in here that I didn't just talk about was number two, stimulus is applied. We actually already kind of talked about that a little bit with the muscle. When, oops, not that one. When acetylcholine binds to its receptor, it opens up a channel and sodium can come in. Okay. When this channel is closed, the membrane's at its resting potential. Okay. When this channel opens, several things can move, but the most important movement is sodium entering into the cell, which is going to start to depolarize it, to get it from negative 70 up. Once it hits negative 55, then the sodium starts to enter through the voltage-gated sodium channels, and you get that very rapid rise in depolarization. But this is the kind of thing that starts it, okay, which we saw at the neuromuscular junction. In other situations, we could see something like this happening, a mechanically gated channel that pops open and lets things in. Now, when I described this, I said some sort of physical stimulus, like the pressure when you're touching your fingertip to something. That'll physically deform the channel, physically deform the cell, which will pop the gate open on the channel, okay? Another example of something kind of like this, which mechanically gated isn't the best example, but does fit with what we're sort of talking about. This could be a thermally sensitive channel, okay? So when the cell is heated up, it pops open and allows things through. Okay? I mentioned that example because when we go here, that's what it would be. The temperature of the water, if this is hot water, if it's a heat-sensitive channel, it'll pop open and let the ions into the cell to depolarize it, and that'll start the action potential, which moves along the axon to the end of the axon. And then a signal is released, neurotransmitters are released that bind to receptors here, causing a new stimulus that reaches threshold. And then we have the action potential go to the end of its axon, where more neurotransmitters released, and that stimulus causes threshold to be reached in this cell, and the signal travels on. And the same thing going back down the other way. Okay. So these membrane potential changes are really what are driving a lot of these. And this applied stimulus part right here, this is another type of membrane potential change. So let me define that too. And that's what we call a graded potential. This is a voltage change in the membrane dependent on the size or magnitude of a stimulus. Okay. So if we're talking about that thermally gated channel, going back to feeling the temperature of water in the shower, the hotter the water, the larger the change in voltage. Okay? And the more lukewarm the water, the less of a change. Now, I'm saying hot water versus lukewarm because temperature sensation is, there's two aspects to it. You have hot sensors and cold sensors. And so it's not that the graded potential that we're seeing in the finger there is based on the temperature of the water, just how much hotter it is than the temperature of the skin normally. The same thing could be said if we were talking about the shower water being too cold. The colder the water is, the greater the graded potential is going to be. The farther it is from the normal amount. We could use a mechanically gated example too. If I'm touching the surface of this table here lightly, then there's going to be a small stimulus, not a whole lot of pressure there. But if I really lean into it, then that's a really strong stimulus. Okay, and so that's going to make for different sized graded potentials. Okay. You could think of it in terms of light stimuli also. A dim light will not make a very big change in the membrane potential of a photoreceptor. And a bright light will make a very big change in the photoreceptors, and then you'll see that. The strong signal from looking at a really bright light means that actually a lot of electrical signals are being sent from your eye into your brain. But a dim light won't send quite the same amount of signal. So those are graded potentials. Oh, another type of stimulus, and actually let me put this here. Types of stimuli. So I suggested temperature. Okay. Hot or cold. Okay, compared to the average temperature at the skin. There could be, I was saying pressure. Okay, so the greater the pressure that you apply as you're holding onto something or pressing into something. What was the other example I was just using? Oh, light. Okay, so brighter light is going to make for a greater signal. Equally, sound is going to make a difference. Okay, so a soft sound's not going to make much difference, but a loud sound will. Should have taken a drink before I did that. Okay, sound, loudness. Taste stimuli will do something like that too. So there are several different tastes, but let's use salty as an example. So saltier foods, I should say, saltier, sweeter, more bitter, sourer, more sour, more sour, or more savory. Those are going to be stronger stimuli. Okay. And then if we're going this way, we'll go ahead and include smell. Smellier is going to be a stronger stimulus. So those are usually some of the major types of stimuli. Okay, just to give you an idea of the kinds of things that can lead to that. The stronger the stimulus is, the greater the potential. Okay. Another stimulus I want to include here is the neurotransmitters. And I want to include that because if we go back here, I was describing a graded potential that we see here at the sensory ending in the skin. There are graded potentials happening at these synapses also, okay? And the stimulus is just the neurotransmitter released from the input axon. So if we have a weak signal out here at the skin, there might be an action potential that travels along this nerve. But when it gets here, if it doesn't release very much neurotransmitter, then it's not going to make a very strong signal on this side. It might not reach threshold. Okay, and same thing here. The signal's not very strong by the time it gets here. It might not be carried on. Okay. This sort of very simple circuit that we see here is going to necessarily suggest that if the signal is strong enough at the beginning, at the sensory receptor, then that signal is going to be strong the whole way through. It's not exactly true, but trying to really characterize that would get into a little bit more depth than I want to get into at this point. Questions about these types of membrane potentials that I'm talking about? Yes.
So, back to the activation gates and the inactivation gates.
Yeah.
So it's at first the activation gate is open at -55.
Okay, and rest, it's the
Right, at
activation
rest.
gate's closed. So it first opens at -55, which I think is what you were saying.
Okay, and the inactivation gate is closed, and then both are open.
No, at rest, the activation gate's closed and the inactivation gate's open. At threshold, at -55, the activation gate opens and the inactivation gate is still open as it was at rest.
Okay,
Okay, so sodium can cross through the channel.
okay,
At +30, the inactivation gate closes. So
okay.
now sodium can no longer get into the cell.
Mhm.
Then during repolarization, when potassium is leaving the cell, the voltage is going to be changing. It's going to affect this, the voltage gauge sodium channel. When it gets back to -55, the inactivation gate closes. Okay? So at that point, both gates are closed
Okay, during repolarization.
during repolarization.
Okay,
And then after the activation gate is closed, the inactivation gate reopens.
okay.
So,
Inactivation gate reopens.
Yeah.
Okay,
but the activation gate's still closed, so sodium cannot cross the membrane.
okay,
So at rest, one is closed. At threshold, they're both open. At the peak, one closes, and then when it passes back over threshold, both of them close, and then
One
back to rest, and one stays open, one stays closed.
all right.
So it's both are only ever open between -55 millivolts and +30 on the upswing.
Okay,
So it opens at -55, activation opens at -55, inactivation closes at +30. That's the only time that the channel is actually going to allow sodium to cross the membrane.
okay.
Otherwise, one, if not both of the gates are closed at all other times during the activation.
Graded.
Yeah.
Graded and action potential, they are different potentials that can occur. Right?
Yes, and graded potentials have various sizes depending on the
With
stimulus.
magnitude. So what action occurs, what action causes an action potential versus a graded, a soft
No,
or,
Actually, after the break, I'm gonna show you a little bit about this, but I'll answer really quickly. From rest, all that can occur are gradient potentials, because the action potential won't actually start until threshold.
Okay.
If the gradient potential is strong enough to reach threshold, then you start the action potential. So that's the stimulus that gets things started.
So it's gradient potential first.
Gradient potential is actually going to be the first thing that happens from rest.
Okay.
So when I was showing you these pictures here, okay, these two pictures, I initially described what was happening with this sort of unlabeled version. And then I switched to this labeled version. And the one thing I hadn't actually talked about in the unlabeled version was the stimulus applied part. And that's the gradient potential there. Other questions? Did her questions there help you understand things a little bit better? Okay. Questions help. Okay. You're sitting there very quietly not asking questions because you might say, I don't want to ask a question. Anna doesn't have that hang up, and Anna just helped all of you understand things a little bit better. If you have a question, ask it, and it's probably a question that somebody else has and will help other people understand what's going on too. So, one more time. Question. Okay. The other possibility is that you're a little dumbstruck and you're just not processing it all. We'll go ahead and take our break.
Yeah.
When we come back at 1/4 by that clock up there, I'll open up with questions. Okay. So if you'd like to, spend your 20 minutes doing whatever, in the back of your head, some of this might percolate a little bit. You might have a question when you come back. Okay, so we'll revisit that question thing then. So be back in here about 1/4 and we'll continue on.